Rockingham Studios presents a Mead Steel Education production. Wild World of Science, Biology, Introduction to Cells. With course instructor Mr. Ronald Jowsey.
Welcome back, one and all. Today we begin a new unit. From biomolecules, we turn our attention to cells. Let's begin by discussing cell theory and cell structure. Cell theory states first that the cell is the basic unit of structure of all living things. This means that all living things are made up of cells. One-celled organisms are known as unicellular organisms, and organisms made up of more than one cell are known as multicellular organisms. If we zoom in on the top picture, we can see some examples of unicellular organisms. These include euglena, amoeba, and paramecium. Now, while the first part of cell theory states that cells are the basic unit of structure in living things, the second part of cell theory states that cells are the basic unit of function in all living things. This means that all of an organism's life functions are carried out by cells or parts of cells. The third part of cell theory states that all cells come from previously existing cells. There are a couple of exceptions to cell theory, though. These are: first, viruses are not made of cells. They do contain genetic material, but reproduce inside another cell, called the host cell. Can you infer what the second exception to cell theory is? For a hint, look in your notes at number 3 of cell theory.
Mr. Jowsey, I have a question.
Okay, Bailey, I have an answer for you. Maybe. Ha ha ha.
If all cells came from previously existing cells, where did the very first cell come from?
Well done, Bailey. And while I can't tell you the answer to that question, I am happy to say that you have inferred the second exception to cell theory, and that is, the very first cell could not have arisen from a previously existing cell. In truth, scientists don't know the origin of the first cell. Now let's take a minute for a quick recap of cell theory. First, recall that the cell is the basic unit of structure in all living things. Second, recall that the cell is the basic unit of function in all living things. And third, all cells come from previously existing cells, not from non-living matter. Moving along in our study of cells, it would be wrong not to discuss how we've come to where we are in the knowledge we've acquired. To that effort, let's take a look at a brief but important history in the study of cells, otherwise known as cytology. Scientists didn't know much about cells until the discovery of the microscope. This is because cells are so small that they cannot be seen without a microscope. Once the microscope was developed, many scientists made important contributions to our knowledge of cells and the nature of living things. One of the earliest scientists to study cells was Anton van Leeuwenhoek. Leeuwenhoek was a Dutch naturalist working in the 1600s. He is credited with the development of the first simple microscope, with which van Leeuwenhoek examined drops of pond water and drew pictures of the organisms he saw. The moina and copepod shown here are two of the tiny water organisms that van Leeuwenhoek most likely saw in his observations. In 1665, Englishman Robert Hooke put two lenses together and made a crude compound microscope. With his microscope, Hooke looked at thin slices of cork and saw tiny box-like structures that reminded him of rooms or cells in a monastery. He called the tiny boxes cells. What he was seeing were actually the cell walls of plant cells. The image shown is what Hooke saw when he looked through this first compound microscope. In 1831, a scientist by the name of Robert Brown conducted his observations of a cell. Brown was the first to notice a small round structure inside of a plant cell. Brown named this structure the nucleus of the cell. With the work of scientists done previously, our knowledge of the cell developed, and cell theory was beginning to become clearer. German botanist Matthias Schleiden studied plant tissue in Germany, and it was Schleiden who was the first to conclude and document that all plants were made up of cells. And then came another German scientist, Theodor Schwann. Schwann was studying animal cells at the time, and he is the first to conclude and report that all animals were made up of cells. Rounding out cell theory, it was Rudolf Virchow, another German scientist, who in 1858 formulated the idea that all new cells must come from other previously existing cells. Now that we've talked about cell theory and its history, I'm hoping you understand that if it were not for the work done by our scientist predecessors and the invention of the microscope, we would not have the understanding of cells and cell processes that we have today. With that in mind, we'll take a look at some of the microscopes used by scientists today. The microscope used most commonly for cell study is the compound light microscope. This is the microscope you will be using in class. For study with this microscope, the specimen being observed must be thin enough for light to pass through it. They are called compound microscopes because they have two lenses or systems of lenses. They are called light microscopes because light from the bottom passes up through the specimen and then passes through the objective lens. The image of the specimen produced by the objective lens is magnified again by the second lens, the ocular or eyepiece lens. You should note, and you will see in our first microscope lab, that as a result of viewing through the compound light microscope, the specimen image is magnified and appears upside down and backwards. The image provided shows a compound light microscope with each of its parts labeled. You will need this diagram when you complete your microscope lab next week. Don't lose your notes, please. Many parts of the cell are almost colorless and are hard to distinguish from other cell parts when viewing with a compound light microscope. Staining techniques have been developed to color certain cell parts so that they are easier to study with a compound light microscope. The most commonly used stains are iodine and methylene blue. Another type of microscope used by scientists is the dissecting microscope. The dissecting microscope is used to study large specimens that cannot be seen with the compound light microscope. It has two eyepieces and two objective lenses. The lower magnification and three-dimensional quality of the dissecting microscope are useful in performing dissections. The dissecting microscope is also called the stereo microscope. The final type of microscope that we'll discuss is the electron microscope. The electron microscope is very powerful and can produce magnifications of more than 100,000 times. Instead of light shining through the specimen, the electron microscope uses a narrow beam of electrons. There is a disadvantage to the electron microscope, however, as specimens to be viewed in an electron microscope must be embedded in plastic and cut into very thin slices. As a result, living specimens cannot be viewed using an electron microscope. The image shown on the right is that of an electron microscope. The image on the left is a photograph of a virus taken with an electron microscope. Another piece of technology that has helped us in our understanding of cells and biological processes is the ultracentrifuge. The ultracentrifuge is used to separate cell parts according to density. The material to be studied is whirled around in a test tube at very high speeds, and the heaviest parts settle to the bottom of the test tube and layers are created according to density. We've discussed that a microscope magnifies the specimen you are observing. Now I would like to talk about the total magnification of what we view using a compound microscope. To determine the total magnification of a specimen under a compound microscope, the magnification of the objective lens is multiplied by the magnification of the ocular lens. There is an example shown here. It's important to note that objects viewed through the microscope are very small and cannot be measured with the standard units of length used to measure larger objects. The unit of length used to measure microscopic specimens is the micrometer, or the micron. This unit is very small. In fact, 1,000 microns is only equal to 1 millimeter. This concludes our notes for today. Please read Chapter 5-1, Introduction to Cells and Cell Study. Highlight important information and vocabulary, and use complete sentences to answer all questions given. Until next time, be safe, make good choices, and study.